Hello physios, in the last video we discussed the anatomy of a human muscle. In this video, we'll understand how a nerve signal is transmitted to a muscle and how a muscle contracts. At the end of this video, I'll let you know easy ways to remember these processes for your examination. Let's start by learning how a nerve signal or impulse is transmitted to our muscles. There are some changes to the axon terminal and motor end plate for this transmission. Let's look at those changes in structure, then study the transmission process. Let's take a closer look at this diagram to study it further. When the axon portion of nerve extends to supply to a muscle, it loses its myelin sheath. See in the diagram, the sheath ends before the axon forms a bulb-like structure. This bulb has two primary components: mitochondria and synaptic vesicles. Vesicles are pouch-like structures that store the chemical acetylcholine. This chemical is synthesized by mitochondria of motor plate and stored in the vesicle. Moving further down in this diagram, you should know that a nerve and a muscle aren't in direct contact but have some distance between two. This is known as synaptic cleft or gutter. So, the nerve membrane before synaptic space is presynaptic membrane, and membrane of the muscle that lies beneath the synapse is called postsynaptic membrane. Besides the nervous membrane and the muscular membrane, the synaptic cleft contains basal lamina, a thin layer of extracellular fluid, and the enzyme acetylcholinesterase. This enzyme plays a crucial role in muscle relaxation. The membrane of a muscle or postsynaptic membrane has folds known as subneural clefts and receptors known as acetylcholine receptors. Now that you know all the specialized features of neuromuscular junction, let's move on to the process by which the signal is transmitted. For easy understanding, we have divided the process into 7 steps. Let's look at each in detail. Step 1. The action potential reaches at the end of an axon and changes the polarity of the axon terminal. Step 2. When the polarity is changed, calcium channels open and calcium from extracellular fluid enters the axon. Step 3. As calcium enters axon, the vesicles move and fuse with the presynaptic membrane where they rupture, so acetylcholine is released in the synaptic cleft.
Step 4. After being released from the vesicles, acetylcholine binds with the nicotinic receptors present in the postsynaptic membrane and form acetylcholine receptor complex. Step 5. Formation of this complex opens the ligand-gated channels. Step 6. From synaptic cleft, sodium enters into the muscle's membrane. Step 7. As sodium starts to enter the postsynaptic membrane, the resting membrane potential of this membrane changes. It goes from -90 MV to -60 MV. This is called end-plate potential. End-plate potential causes development of action potential in the muscle. Step 8. Destruction of acetylcholine. Acetylcholine is so potent that the short duration of 1 millisecond is enough to excite the muscle fiber. To prevent repeated excitation of the muscle, and allowing relaxation after contraction, this acetylcholine should be destroyed. This task is accomplished by the enzyme found in the synapse, acetylcholinesterase. This enzyme breaks acetylcholine into inactive choline and acetate. Choline molecule is taken back into axon terminal and reused by the vesicle to form new acetylcholine molecule. Now that the action potential is generated, meaning the muscle is excited, let's see how it contracts. As action potential is generated at a site in the muscle, it travels all over the sarcolemma through T-tubules. T-tubules are in contact with the muscle fiber and cisternae, or ends of L-tubules. These cisternae excite as action potential reaches them, leading to the release of calcium ions into sarcoplasm. This calcium moves to actin so that muscle contraction may begin. Calcium ions bind with troponin C and produce change in its position. Changing position of troponin pulls tropomyosin, and, with the movement of tropomyosin, the active sites of actin are uncovered and exposed. As the active sites are exposed, immediately the myosin head attaches. After attaching to actin, the myosin head tilts, which moves two actin chains closer to each other. This is called as power stroke. Energy for the tilt of myosin head is brought by breakdown of ATP into ADP and inorganic pi by the enzyme ATPase, which is present in the myosin head. After tilting, the bond of myosin head and actin breaks and myosin head returns to its position. Next, it binds to new site in actin, tilts back from the energy of a new ATP, followed by breaking away from active site of actin. As this process happens again and again, the thin filaments is pulled towards the center of sarcomere. When opposite side actin filaments overlap, we'd call it actin-myosin complex, or the contraction of muscle. When the muscle contracts, the H-zone narrows and disappears, and Z-lines come closer to each other. This theory of muscle contraction is called a sliding filament theory, or Ratchet theory, or walk-along theory. For your examination purposes, you can screenshot these flowcharts as they'll make the process of muscle contraction easy to remember. That is all for today. If you have any questions, post them in the comment section. I'd love to hear and answer them. Like, share and subscribe if you understood the content.